51 to 60. So before starting our project, we want to say the case scenario, which is a 2-year-old male boy who was having brittle hair and delayed developmental milestones and was having mental retardation. And the homocysteine was having, in the plasma was 105 micromolar per liter. The normal value should be around 5 to 15 micromolar per liter. And the cyanide nitro peroxide test is positive. So the diagnosis is homocystinuria. Now giving to Nandana for the normal metabolism of homocysteine.
This is normal metabolism of homocysteine. Here, methionine is converted to homocysteine. Homocysteine, in the presence of serine, with the help of cystathionine beta-synthase is converted to cystathionine. Cystathionine, with the help of pyridoxine phosphate and cystathionase enzyme, is converted into cysteine plus alpha-ketobutyrate plus ammonium ion. If this cystathionine beta-synthase is absent, then type 1 homocystinuria is found. I'm handing over to Nilan.
For methionine to be regenerated, homocysteine should be methylated. Methyl group is derived from folate. Methylene tetrahydrofolate is converted into methyl tetrahydrofolate by methylene tetrahydrofolate reduction. Methyl group to methyl tetrahydrofolate is transferred to vitamin B with the help of methyl tetrahydrofolate homocysteine transmethylase enzyme. And from methyl B12 methylate homocysteine into methionine. The deficiency of methyl tetrahydrofolate detected causes type 2 homocystinuria. The deficiency of methyl tetrahydrofolate homocysteine transaminase transmethylase causes type 3
Let's talk about the functions of homocysteine and cysteine. First of all, homocysteine and cysteine are non-essential amino acids which are glucogenic in nature, and they're mostly present in the keratin of nails and hair. And the main function of homocysteine is it helps in the synthesis of cysteine. And cysteine as a part in the membrane, maintaining the membrane integrity of the RBCs. It helps in the synthesis of glutathione, and moreover it helps in the transfer of amino acids. And let's come to the function of methionine. Methionine, it's mostly related to methylation reactions, and it helps in transfer of methyl group. For example, it helps in conversion of noradrenaline to adrenaline. I would like to hand over to Praveen.
Then in the explanation part, let's get into the action part, where we use this contraption-based model to help us understand homocystinuria much better. We test this model. First, this is the chromosome and carrying the 21st gene, which carries the gene for the homocystinuria. The balls represent mutation. When I just plug out this gene, the balls roll out, initiating a reaction where these reactions, which are supported by the respective enzymes, cystathionine beta-synthase, methyltransferase, and methyltransferase reductase. In a normal physiologic condition, in our bodies, homocysteine is converted into cystathionine and methionine as well, under normal conditions, with the help of these enzymes. But in abnormal cases, where under mutation, these enzymes become deficient and the reaction bridges are broken, which will be represented by pulling out this plug. Then the metal balls which we placed inside represent the homocysteine, which accumulates and enters into the bladder, which are unfiltered by the kidneys, which sets up another reaction to show the clinical manifestation of the homocystinuria, which is ectopic dermatitis, dislocation of the iris. So let's start the reaction. As you can see, the iris is dislocated. Also, these vitamins act as a cofactors for the reaction to succeed. On the deficiency of these vitamins, for example, in the case of folate, which is the cofactor for methyltransferase, and vitamin B12, which is the cofactor for methyl reductase. Upon either the enzyme deficiency or vitamin deficiencies, these homocysteinurias 1, 2, 3 are manifested.
So symptoms of homocystinuria is first eye lens dislocation, which is due to weakness of glomerular fibrosis, which is present in the eyes. So next is that pave travas, which is the arch of the foot won't be as normal; it would be more arch. And the second is tall stature and a high arch barrel, and main is osteoporosis. These are the common symptoms of homocystinuria, and I will pass to Asif for the more information of homocystinuria.
And the people not born with this are the people who got alcoholics and smoking and post-menopausal women. If a pregnant lady got this disease, her child, there's a great chance that her child should aim and develop a proper neurology. And there's a great possibility that the patient will going to develop coronary artery disease and Alzheimer's and other kind of disease as well.
Now we are going to see about the biochemical diagnostic test. In order to detect and confirm the homocystinuria disease in some individuals, we have to perform some of these tests. Like for homocystinuria type 1, cyanide nitroglyceride test is a test which is a simple quantitative test which is performed in the urine sample to detect the quantitative amount of the amino acid present in the urine. And some of the other tests are also done, like the Guthrie's test and amino acid screening, liver biopsy, and finally to detect the whole disease we do genetic level testing. Now I hand over to Amina. At last, coming to the treatment and prevention. Treatment of homocystinuria includes special diet. Special diet includes increase the amount of cysteine and decrease